Hey guys, this is Dr. Hub. Today we'll look at the enzymes and the enzymes which are involved in the DNA replication. So to begin with, first is the DNA polymerase 1. So in short, it is known as a POL1. So what is the function of this? Here there is a gap filling followed by the DNA replication. So there is gap filling following the DNA replication. So following the DNA replication as well as the repair and the recombination. So there is repair as well as the recombination. Next we move on to the DNA polymerase 2. The DNA polymerase. The DNA polymerase 2 is also known as the POL2. So the function of this is that the DNA proofreading and the repair. So it does the DNA proofreading as well as the repair. So it is associated with these. So 2 is associated with two functions, that is a proofreading and the repair. Next we'll move on to the DNA polymerase 3. So in the DNA polymerase 3, there is it is also known as a PRO3, POL3, and this is associated with there is a processor and the leading strand synthesis. There is a processor and also the leading strand synthesis, leading strand synthesis, as well as there is a DNA proofreading. There is DNA proofreading. Next what we have is the DNAG, and DNAG is the function is nothing but the primase. Next is the helicase. The helicase, the function is nothing but it is a ATP driven processor, unwinding of the DNA. Unwinding of the DNA. Next we move on to the primase. So what is special of the primase? The primase, it initiates the synthesis of the RNA primers. It initiates the synthesis of the RNA primer. Next is the topoisomerase. The topoisomerase. So topoisomerase is nothing but it relieves torsion strain. It relieves torsion strain, and that results from the helicase induced unwinding. So it results in helicase induced unwinding. Next we move on to the single strand binding proteins. Single strand binding proteins. So this in short is known as the SSBs, and what this does is it prevents the premature reannealing of DNA. So it prevents the premature reannealing of the dsDNA. Next we move on to the DNA ligase. So in the DNA ligase, it seals the single strand nick. It seals the single strand nick, and this is between the nascent chain and the Ozaki fragment and the Ozaki fragment. So it is between the nascent chain and the Ozaki fragment on the lagging strand. On lagging strand.